Hey everyone, my name is Daniel Gataolen. I'm one of the senior research analysts covering biotech here at Chardan. It is my pleasure to introduce Shankar Musunuri, the CEO of Ocugen, to our second annual Virtual Ophthalmology Summit, where the focus of today's discussion is on AMD and diabetes-related ocular diseases. Shankar, welcome.
Thank you. Thank you, Daniel. Thank you for having me today.
Of course. So to get things started, could you please spend a few minutes and introduce yourself and Ocugen to the audience?
Absolutely. I'm Shankar Musunuri, Chairman, CEO, and co-founder of Ocugen, 30 plus years veteran in biotech industry. Ocugen is a biotech company focused on primarily gene therapies targeting ophthalmology diseases. We have three first-in-class platform technologies, starting with modifier gene therapy platform targeting ophthalmology disease area. We have first-in-class platform technology for knee cartilage repairs. It's a NeoCart. That's phase 3 ready asset. And we also have inhalation platform technology targeting COVID and flu. And so with that, we are basically targeting rare diseases with our gene therapies, as well as diseases that affect millions, like AMD.
Excellent. Thank you, Shankar. Let's talk about your gene therapy programs in ophthalmology. So you're using a very differentiated approach, modifier gene therapy. Can you explain what that modifier gene therapy approach is and how is it different from a more traditional gene replacement therapy?
Yeah. So the traditional gene therapy, Daniel, I think well, let's take a genetic defect, and when you have disease like retinitis pigmentosa, about defects in about 100 genes can cause it. If you take a traditional gene therapy approach or gene editing, you can go one gene at a time. You cannot go more than that. So disease like RP requires 100 products. And with the modifier gene therapy approach, what we are finding out, when you have a defective gene, genes work as a network. They don't work in isolation. So in our human system, there are more than 30,000 genes, functioning genes. There are 10% of them in our eye system. And so they all work as a network. And when you have a defective gene, so the network is also kind of corrupt. So you have a depression of this modifier or master regulator genes. And these are like a transcription factors, what we have NR2E3, which we use for, it's a master regulator, we call it modifier gene. So it is also depressed. So when you give, this master regulator, so it has ability to control the entire network from cell development, inflammation, metabolism to survival. And it resets the homeostasis at a molecular level and cellular level. That means several, genes which are needed to function well, they're also depressed because the transcription factor itself is depressed. And so once you give this, it resets the homeostasis and restores the function, and also creates a healthy environment for cells to survive. I mean, in this case, photoreceptors, if you reset them, if the cells to survive, that's really important for, protecting or, vision what you have, or in case, just like we saw in our phase 1/2 data, in some of the patients, when they're losing the peripheral vision, it's coming back. And so I think it's really important to understand. So in case of traditional gene therapy, you go like one gene at a time. In some cases, because you got a problem with the entire network, you need to think about holistic approach. And sometimes, with the traditional approach, you may not even get the optimum results you're expecting because, you have the whole system is corrupt. And how do you fix it? One has to look into that. That's where modifier genes really look at, looking at the entire network and how do you bring homeostasis and restore function along with, creating healthy environment for these cells to survive. That's very important.
Got it. Understand. Thank you. And so you're applying this modifier gene therapy approach for both rare diseases such as retinitis pigmentosa, Stargardt disease, but you're also applying it for a more prevalent condition such as dry AMD. So is the focus of today's discussion on AMD. Can you talk about why you believe your asset for that indication, OCU410, is a promising solution for dry AMD patients. And maybe to expand a little bit, how does OCU410 contrast with currently approved therapy uh, for that indication?
Yeah, absolutely. So AMD is a very complex disease. When you take dry age-related macular degeneration, there are many pathways which result in the disease, starting with oxidative stress and lipid metabolism. The key factors, as we all age, obviously, you need certain antioxidants, and you have lipids like lipofuscin, which are needed for function. And the lack of, the effect of that, they'll create what you call it, at a very tiny level, molecular level, aggregates. And our system, we have RPE layer, which is supposed to clear it and recycle. When the system doesn't work well, you get inflammatory markers like cytokine reaction, right? And that results in AMD. And the final pathway is the complement system. And so obviously the two products in the marketplace, approach has been working on the final outcome, which is a complement system. The formation of that causes degeneration. However, people really have to look into disease pathology, how the disease starts because of oxidative stress and lipid metabolism. And one has to really look at how do you control all these pathways, regulatory pathways. So the two products in the marketplace, the facts are there. The data is there. You have to take 6 to 12 injections per year, and they have a compared to untreated control in phase 3 clinical trials, they showed they reduce the lesion growth by 30% in 2 years. And that means that atrophy area, the lesion is still growing with the treatment. It's going at a lesser rate compared to control. And also in one of the products, they showed about 12% actually result in wet AMD for treatment with the dry AMD. That means after 2 years, that's also a safety concern, right? I think if you take those factors, it's a compared to control arm, you're seeing a decrease in lesion growth because there are no therapies out there. That's okay. And so now you park that idea. Now you come back how our gene therapy works. And obviously there is a RORA gene, which is targeted for AMD. And we have shown with our in vitro, in vivo studies, demonstrated with the strong data that it has ability to regulate all 4 pathways.
Got it.
That's very important. And so that I think going to the core idea of oxidative stress and lipid metabolism, inflammation, and blocking the complement system. So when you regulate all that, that's really important. And so you expect it to perform well, as we have shown in our in vivo data in the animal studies, reduction in drusen. And also, we don't give CD59 expression like other companies try to target for a complement system. And CD59 expression in that specific animal model we showed is missing. And that's, again, a modifier approach. We take a ABCA4 defective model. And the ABCA4 defect, ABCA4 defective gene, CD59 expression is depressed. Now I'm going back to network, right? And why is CD59 depressed? So when you have a defective gene, it impacts, these transcription factors and all underlying, all functioning genes. Many of them can be depressed too, which are needed for function. So in this case, CD59 is a great example. And we're not giving gene therapy for CD59 expression. We're giving our RORA in this model. And we showed CD59 expression comes back, just like how it's supposed to be. So that's, again, showing all 4 pathways, demonstrating in vivo, in vitro models scientifically that, we regulate all 4 pathways. We believe this is the way to, treat this disease with a single subretinal injection for life, potentially. That's what we have compared to what is in the market today.
Got it. And how is 410 administered?
It's a subretinal injection.
Got it. Okay, so you talked about RORA and some preclinical models. What specific models did you use, and is there any clinical support, in addition to the preclinical data that you described that gave you confidence in the program?
Yeah. So I think what we did is we started with models such as ABCA4 defective gene model. That's the animal model we use for dry AMD as well as Stargardt disease. As I explained before, in that model, we showed CD59 expression is almost very low or missing in the defective these animals. And when we give RORA, it shows the modifier approach and working on the system, and you get the CD59 levels back. We also looked at the fundus imaging, looking at the lipid metabolism causing the drusen, these particles, and the drusen significantly getting reduced compared to untreated animal models. And we also developed cell culture models for showing anti-inflammatory properties of RORA significantly. And we also have a oxidative stress model. In that model, we looked into all our modifier genes. NR2E3, which is for RP, NR1D1 is another modifier gene, which works on circadian rhythm. And also we have RORA. And RORA actually showed significant antioxidative properties in that cell culture model. So putting all this together, it again demonstrates there are 4 significant pathways that cause dry AMD, and it's important to really address all of them and regulating these pathways. And that's why we believe RORA is a powerful modifier gene which has the ability to regulate all these pathways and bring this system together so that it has the ability to potentially control significant decrease in the lesion. And it has potential to provide, clinical data will tell again, or we are still in the phase 2 clinical trial right now. Our phase 1 data from dose escalation study, including preliminary efficacy, we're anticipating by the end of the year. And so in that, obviously, what we're going to look for, just like in the commercial products, you look for the lesion size, from the baseline. How is it growing? And how our therapy is working out, based on some data which is available, natural history, and the products which are already in the market. Obviously, the phase 1 trial doesn't have a true untreated arm, which is part of our phase 2. And so we had to do some measurements based on natural history and also a couple of products which are in the marketplace, do some extrapolations based on, typically untreated populations in these clinical trials. What is the decay and the growth of the lesion size on annual basis, and how do we compare without treatment?
Got it. Understand. So as you just explained, OCU410 is in clinical trials now. So the dose escalation portion is complete, right? You said data by the end of the year from that portion. And you're enrolling the phase 2 portion. Can you walk us through the trial design in terms of the trial size, the duration, and what target population are you looking at?
Yeah. And so what we are looking for is, again, in the late stage kind of dry AMD, which is called geographic atrophy. And obviously, the inclusion criteria will be they need to have a certain lesion size, from 2.5 to there's a number next to it, so it has to fit in that atrophy area has to be targeted. And in phase 1, what we did, we did the traditional dose escalation study, 3+3+3, low dose, medium dose, and high dose. And obviously, the safety and tolerability looks good. And so that's why we took like 2 doses from the phase 1 and moved into phase 2, the medium dose and high dose, and along with the untreated control arm. So the phase 2 will have 15 subjects in each arm. So you have medium dose arm, high dose arm, and untreated control. It's SSR blinded study. And once again, what we're looking for is that structural lesion, along with the safety, we'll be looking at the efficacy based on that, because that's what the approved product used, structural endpoint, measuring the lesion. And so that's what we're going to monitor on the patients compared to untreated. We'll have at least untreated arm in our own clinical trial.
Right.
stop using the natural history, which will be really good. And it's a 1-year duration. And so we'll be doing the phase 2 after recruitment. We'll monitor the patients for 1 year. So that's the plan. In addition to the lesion size, we'll also look into some other metrics, sensitivity in that retinal area, looking at microperimetry. And also we can look into BCVA and LLVA compared to untreated arm. In addition to that, there is also quality of life measures, we're going to follow through. So because phase 2 is all about, look at what FDA approved, what is the endpoint, how do we do that? That way we can do the comparison, right? That's really important. In addition to that, are there any functional measures we can look into, and quality of life measures? And those are important too, to monitor in these patients.
Got it. Got it. Okay. And when you report, your initial data by the end of this year, and later from the phase 2, what results would you consider a home run? What would make you happy when you summarize those results?
So I mean, obviously, with the limitation, right? At the time is important, that duration. And so, I mean, typically, those companies took almost 2 years to show the data and to get approval. So what we are looking for this year is, at least in our gene therapies, the way modifier genes work, in traditional gene therapies too, it takes some time, unlike small molecule biologicals, and in those cases, you get a pharmacodynamic effect right away. With gene therapies, it takes time, because especially with the modifier genes, the network has to work. So what we're anticipating is at least a few patients who complete 6 months from early phase 1 clinical trials, low and mid dose. And obviously we're going to look for a lesion growth, from the baseline, and obviously we're going to compare based on some data which is already in the public domain. And are we significantly reducing the growth within the time frame compared to what is out there? That's number one. And if you're significantly reducing it, and you're also showing any other quality measures, including some functional improvement in these patients, and with a single potential subretinal for life instead of taking 6 to 12 injections, that for me is a people don't have to take 6 to 12 injections with good safety. So far, the safety looks good, and so that's as I mentioned before, they have about 12% getting into wet AMD. That's a safety concern. And if we can show a single therapy and significantly reducing the lesion growth with a single injection with a good safety data, that's a home run. And obviously the gene therapies we showed in our first program have ability to even stop the growth and reverse it in some patients, right? That's what we're seeing improvements in the first program. Similar to that, they have potential. Once again, proof is in the pudding, so we need to look at the clinical data. And these genes are very promising. And we're hoping, that if we can give an alternate therapy, which has a single dose for life, and it is significantly reduce the lesion growth, and it also shows some quality measures. And that's what patients need.
Yeah, got it. What are you hearing from various KOLs and patient groups regarding the need for a new treatment for dry AMD?
I mean, we have a lot of good centers with very highly credible retinal surgeons doing our clinical trials, for our AMD program. And one of the we get a lot of calls from patients who are on current therapies in the marketplace, and the compliance, taking every month or every alternate month injections, and not ability to show or feel some functional improvement. And those are the critical things I think patients are looking from. And from that perspective, we do get calls. Obviously, we can take the patients. There is a cool-off period, several months. I think they have to pass that, and they'll be qualified for our clinical trials. So we will take those patients, but there is a cool-off period. If you're in any of those market products, we need, I think, minimum 90 days. I need to confirm that, but I think, yeah, after that, if they're qualified with our inclusion exclusion criteria, and we'll take them into our clinical trial. So, I mean, compliance is one, and I think people are expecting, definitely probably looking for better improvement than what the current drugs are doing in multiple ways. And so that's why we really want to focus on that.
Got it. Okay. Understand. And another question that I have on 410, given its very different mechanism from the current approved medications, do you think there could be a synergy of using it in combination with other drugs, or do you think it will be a standalone therapy, or is it too early to tell, and that decision will be driven by the data that you generate over the next months and longer?
Yeah, I mean, obviously, our trials are going to be like a monotherapy with our gene therapy. And once again, we have to see how the market evolves in the next few years, because if we can show either significant reduction in the lesion growth with some quality parameters and some functional improvement compared to those therapies out there, I mean, how it's going to impact. If you have therapies out there, which are good in short term, just controlling something, if you have a disease, and then you have gene therapy to take it to long term, that's an option as a combo. But here we have to see that if they significantly do something, but they're not good for long term, but you need frequent injections, right? And if I mean I would use similar to wet AMD or DME treatments, right? And if you give any anti-VEGF, we know it can control the leakage very shortly, and obviously then you can do something else above that, if you want a long-term effect. So I think we have to see. And obviously, our focus is going to be monotherapy. We believe if our therapy has potential to treat these patients with a single injection, and if that gives optimum results, what patients are looking for, probably you don't need anything else. So
makes sense.
That's going to be our focus.
Makes sense. Okay. And another question on manufacturing. So I know you are completing your manufacturing facility with gene therapy production capabilities. Can you talk about that facility, and will that be providing the clinical and then in the long run commercial product for OCU410?
Yeah. So we have a for OCU410 product, all our gene therapy, we have a ex-US strategic partnership, and we have plenty of commercial capacity from there. However, we also built facilities next door in our Malvern, Pennsylvania headquarters, where we are, and that has a good capacity for our gene therapies for US market. And our goal is to get that up and running in the next couple of years. And obviously, we don't want to change the train right now for the first program because we're already in phase 3. So we cannot make any CMC manufacturing changes because we produce a very high quality product. Everything looks good. And obviously, for the first program, that will be added, after the BLA approval as an additional site and our backyard manufacturing in US. And for the second one, that's a great question. Currently, we're in phase 2. We got another year to go. And so before that time, we'll make a decision, for phase 3 for US, should we use product produced in our shop here, right here, next door in US versus, should we use the material from ex-US partner? That's very critical because FDA, expects the phase 3 for gene therapies to be at commercial scale, which we did for our first program. So you minimize any risks, and then the facility and everything in the process are already, well defined before you get into phase 3, so that you don't have many changes after that, so that the CMC and manufacturing is robust. So that decision, we have time to do that. We're not worried about capacity. We have capacity here, the way we built it, and we got plenty of global capacity elsewhere with our partners. So that I think we're going to weigh those options in the next 1 year. And if we decide for phase 3 and we need our facility, then we have to get it up and running. And so, before we start the phase 3.
Got it. Understand. Okay. So in the next few minutes, I wanted to talk to you about your other program. It's a biologics program, OCU200 for DME. Can you tell us a little bit about this drug candidate and what differentiates it from a number of other approved and development candidates for DME?
Yes, yeah. Wet AMD and the DME, this is a very established market, and that's the reason, there is a lot of attention. Whereas dry AMD market is still getting established, and I think people are getting to know it's good. There are two products in the marketplace. They're educating, patients and physicians, everything else. So here the landscape is already set and it's growing, right? With the diabetic population, with the diabetic retinopathy as well as with AMD. So here, most of the products, the first line, they target anti-VEGF therapies like Eylea, and there's a bifunctional antibody from Roche, and their main target is anti-VEGF because you got a leakage because unwanted, blood vessel growth, and the leakage needs to be stopped. So your vision immediately, you'll see the improvement. And so those anti-VEGF therapies are good. However, there is a significant number of patients who may not respond to those therapies. That's missing. They still don't have an answer for those patients, right? And so what do you do with those patients? Number two is that approach we're taking, including gene therapies targeting that, as a delivery system, not a genetic approach, using instead of anti-VEGF injections, taking like either 4 times, 5 times, 6 times a year, can we put a gene therapy vector, so we can continue to release this anti-VEGF at a lower level. They may still need some interventional, medicine such as anti-VEGF on a periodic basis, but at least can they release something at a certain level so they can minimize those number of injections. So the whole market is anti-VEGF and reducing number of injections. They're not looking into those patients, who are not responding to those therapies. And also, as you know, VEGF is also needed for, healthy cells, for normal cells too. And the blocking is one thing, and, so you need to balance that scientifically. And so where we come in is we have a tumstatin transferrin. It's a fusion protein, and it's a one of its kind. It's a large biological. We have good in vitro, in vivo data, head-to-head comparing with Eylea. And we believe tumstatin targets active endothelial cells and integrin receptors, which are present on it. So it doesn't touch the normal cells. From safety perspective, I think this is good. And we also showed at very low concentrations, head-to-head models, it's doing well in vivo, in vitro studies we did compared to existing therapies out there. And so by targeting active endothelial, number one, number two, this has a potential to be disease-modifying therapy. Number three, it can also, based on the mechanism of action, target those non-responders who are non-responding to current therapy. So I think those are the salient features with this molecule. And also we have a transferrin fused to that will help to transport this molecule to the proper site and target, and also improve the uptake. So that's why it's effective at a low doses based on our data we generated from in vivo studies. And so where we are with that, that molecule, we believe is needed for these patients for the reasons I stated. Number one, it's a distinct mechanism of action. It has potential to be a disease-modifying drug, not just targeting anti-VEGF. And then also ability to have the safety because it doesn't touch the normal cells. It only targets active endothelial cells. And also you got significant non-responders, which you need to target too. And we believe this has potential to work on entire patient population, not just the responders of anti-VEGF.
Got it. Okay, I understand. And so in terms of the status of that program, so you filed an IND last year, I believe, but it was placed on clinical hold pending some assay validations. Can you give us the status update of where you are in the process of addressing the FDA's questions?
Yes, our team is in fact working on addressing those questions, and they're anticipating to submit responses this year to satisfy FDA. And after that, once we get the clearance, the program as a phase 1 study, which is already planned and designed. And it's a dose
Okay.
escalation, just like, anti-VEGF therapies. And then the good thing about the DME or wet AMD in this disease area, because of the usage of, these medicines which are available for many, many years. And I think the retinal surgeons who do these clinical trials, they anticipate results sooner than later, even with a phase 1 clinical program. And so I think we're excited about it, the program. I think there's so much of underserved population within that, disease area. So I think we will work, continue to work with the agency, satisfy their questions, and hope to get that clinical hold cleared and get into the phase 1.
Got it. Okay. And so taking funding commitments for your gene therapy programs into account, what's the current strategy for OCU200 once the hold is lifted? Do you plan on beginning dosing patients right away, or is that going to be second priority, and then you're going to be looking for a partner as you advance your gene therapy programs?
Yeah, good question. Our focus has been, for gene therapies, because we have 3 programs in the clinic, 1 in phase 3, 1 in phase 2, 1 in phase 1. And obviously, we believe phase 1 for OCU200 is relatively not that expensive, because what we are thinking is, at least in order to partner that asset, definitely we would like to partner that, at least get the preliminary safety and efficacy data in the clinic. We anticipate results sooner than later compared to gene therapy. So once we get to that stage, that will be efficient way of doing that. And then we look for partners, because it's a traditional biological. So by the time you go to phase 2, it's going to be a big program, and then phase 3 will be even large compared to our gene therapy. So we definitely, it's a very valuable asset, and it's a very established, market, and that probably will attract a lot of large pharma companies to partner that.
Yeah, understand. And just a couple of questions to conclude the session. What is the current operational runway, and what assumptions are you making?
The current runway is obviously, we're going to continue all our clinical programs, which are in the clinic, and we're going to get to 3rd quarter next year with the recent rise we did. And the reason we did that is, to make sure we're not rising too much and we are sensitive to our current shareholders and dilution. And we also wanted to make sure we do a good fundraise, which has a minimal effect on our current shareholders, and also extend the runway. I think that'll give enough room for us. The reasons are, as we talked on this call, we're anticipating some preliminary efficacy data from our preliminary phase 1 on our OCU410, and which people are waiting for that, and there are potential partners who are interested in that. And in addition to that, we're also having discussions on OCU400, regional partnerships or global, looking into various reasons. So we want to wait how these things will materialize in the next 6 months, and based on that, if we get a potential partnership, that gives us a non-dilutive funding, and that can extend the runway further, because we are currently we're managing very efficiently the operations of Ocugen, about 12 million a quarter. So I think our ask is not that too high compared to a lot of biotech companies out there, especially considering we have 3 gene therapy programs, and also we're very efficiently run, and therefore we're going to carefully assess early next year. And if we do have potential partnerships through upfront, and we get some significant upfront, and that will help us to minimize any further fundraise and dilution. So we're going to balance all those and carefully assess, and as needed, we'll continue to raise money. And so that's the plan.
Okay, makes sense. And one last question before we conclude the session. What do you think investors are missing about Ocugen's story?
I think the number one thing is we really want investors to understand, even though we have other first-in-class platform technology, our focus has been gene therapies for the last 2 years, and it will continue to be gene therapies. what we are doing with other assets, as we have shown in the vaccines, we're partnering with the government, and they're actually going to sponsor and do those clinical trials. And similarly, we're going to find potential opportunities for those first-in-class platform technology to fund those programs. And so that, we're not kind of diluting our effort from capital perspective. And we want to create value for our shareholders. They're phenomenal technology platforms. We'll find ways to creatively, find funding for those through partnership opportunities or some other means. But our focus is continue to be gene therapies. It'll continue to be gene therapies. We want to be gene therapy leaders in ophthalmology. And, our genes can go beyond ophthalmology area once we get some success here. And that's our focus. That's the message I want to give to investors.
Got it. Excellent. Well, thank you, Shankar. I believe you're out of time, but again, really great to chat with you, and I look forward to connecting with you again soon. Thank you.
Thank you, Daniel. Thank you for having me again.